Hello everyone, and thanks for logging in today to the first in our new webinar series, Introduction to Flow Cytometry. My name is Paul Lohan. I've been with MSC for 2 years, and before this I did my PhD and postdoc in NUI Galway, and among the techniques I have experience with are flow cytometry, cell culture, and ELISA. Lisa Tang is the Life Science Sales Manager. Lisa got her PhD from the University of Leeds and has 7 years of postdoc experience. Her expertise is in molecular biology, protein expression, and Western blotting. Emma Fitzpatrick joined the MSc Life Science team earlier this year after about 2.5 years of postdoc experience, and she has expertise in cyto and histochemistry, flow cytometry, and Western blotting. So today we'll cover some of the basics of flow cytometry, from the principles of how the technology works, how to prepare your samples, how to stain and fix your cells, how to set up the cytometer for an experiment, data analysis, and finally we'll have a look at some questions and answers at the end. So many companies have many different models of flow cytometer, and these can vary widely from small compact units with 3 or 4 colors to large analyzers which can handle over 20 colors. There are also sorters which can isolate very specific populations or single cells, and there are even imaging cytometers which can combine the throughput of flow cytometry with the in-depth information of single-cell microscopy. Possibilities are vast, but whatever cytometer you end up using in the lab, the basic principles are largely the same. As well as this, the reagents you can buy are generally compatible across many different cytometers. So, before we jump into the technical details, we should define what flow cytometry is and what it can do for us in biological research. So flow cytometry is a technology which can simultaneously measure multiple physical parameters of single particles. So what do we mean by physical parameters or physical characteristics? Well, flow will give us information on the size of a particle, the granularity of the particle. This is information on the internal complexity of the particle, and it's particularly useful for different cell types where they might be of a similar size, but the granularity can give us really useful information on the differences between them. An example of this is if you're looking at, say, the activation of macrophages, where the cells become more granular as they're activated. And of course, as we all are probably aware, flow also gives us information on the fluorescence signal from each particle. Now, we'll just move on to talk about single particles. So this might seem obvious to us. We're speaking about cells. And yes, most of the time, the particles you will be measuring by flow cytometry will be cells. And that these could be cells from different sources, like cell culture experiments, from organs or tissues, or even blood from donors or patients. You could be looking at microorganisms or even whole animal digests, small animal models like Hydrotinia or Drosophila. Other than cells, some really useful information can be gotten from using beads in flow cytometry, and there are many ways beads can be used. For example, polystyrene beads can be conjugated with antibodies, and these can be used to bind extracellular vesicles or exosomes. And there are also the platforms using fluorescent beads, such as the Geniplex, which is essentially an ELISA that it can be run on a flow cytometer. So now we'll look into how a flow cytometer gathers the information on your particles. Now I'm not going to go into very, very fine detail, but I will sort of scratch the surface so that you have an appreciation for how the cytometer works on the inside. So the first internal part of the cytometer we'll look at is the fluidics. The purpose of the fluidics is to transfer the particles in a fluid stream to the laser for interrogation. To get the best result, the stream should be centered in the center of the laser beam, and only one cell or particle should move through the beam at any given moment. So how does the cytometer achieve this? Well, the sample is injected into a stream of sheath fluid, which you can see here is the sheath fluid, inside the flow chamber of the flow cell. So the flow cell is designed so that the sample core, the part where the cells are, is positioned in the center where the laser beam then can interact with the particles. Because of laminar flow, the sample core and the sheath fluid don't actually mix, but the flow of sheath fluid accelerates the particle towards the laser and restricts them to the center of the flow. And this is known as hydrodynamic focusing. In most cytometers, you can change the flow rate through the flow cell. A higher flow rate will gather data from more cells in the same amount of time, but you lose some resolution. So cells may be less aligned to the center of the beam, and this is quite okay for larger phenotyping experiments. But if you're doing, if you require high resolution, like in DNA analysis, you may need a lower flow rate, which means that most of the cells pass through the center of the beam and they're quite well aligned, but less cells go through per unit time, and therefore it is slower. Of course, there are other methods of getting a stream of single cells through the laser, like acoustic focusing, but I won't go into that today, but there are cytometers that use acoustic focusing as well. So next we need to talk about scatter. So light scattering occurs when a particle deflects laser light. So how the particle scatters this light can give the user lots of useful information on the physical properties of the particle, for example, its size and its internal complexity. The first type of light scatter we'll talk about is forward scattered light. So forward scattered light is light which passes by the cell or is diffracted slightly by it. The light moves past the cell and is detected by a detector, which is directly in line with the laser source. Forward scattered light indicates the size of the particle or cell. The second type of light, which we'll talk about, is side scattered light. Of course, we all know that cells aren't solid, they're not uniform particles, so the light which doesn't pass through to the forward scatter detector is not reflected straight back at the laser, and it's not absorbed. Instead, it bounces off organelles or granules inside the cell and reflects sideways, where it can be detected by side scatter detectors. And this gives a side scatter signal, which is an indication of the internal cellular complexity. Now here we can see a practical example of this from an experiment. So we see side scatter here on the y-axis, and we have forward scatter on the x-axis. You can see that by using only forward and side scatter information, we can separate these cells into lymphocytes here, which have a low side scatter signal and a relatively small size. We have monocytes here, which are larger than the lymphocytes, but they're not too granular. As you can see, they have a low side scatter. And finally, at the top, you can see the large granular neutrophil population. So now we'll talk about fluorescence. A fluorescent compound is one which absorbs light over a wavelength range which is characteristic for that compound. This absorbed light causes an electron in the compound to be raised to a higher energy level. The excited electron is unstable at this higher level and quickly releases the energy as a photon of light and reverts back to its base level. The released light is known as fluorescence. So as we said on the previous slide, fluorescence results from a molecule absorbing light at a certain wavelength and releasing it at another wavelength. So here you can see this shown in graphical form. So we can see the absorption spectrum here on the left, or the excitation spectral profile. And this is the wavelength of light at which a molecule absorbs light and its electrons become excited. Similarly, we have an emission wavelength, and this is the wavelength range over which the molecule releases light. So the emission spectrum is always at a wider wavelength than the absorption spectrum, which you can see here as the wavelength is notated in nanometers. So how do flow cytometers make use of fluorescence? Well, there are two parts to this, lasers and optics. And first, we'll talk about the lasers. So laser stands for light amplification by stimulated emission of radiation. And basically, a laser is a focused beam of light of a specific wavelength. Flow cytometers will come with one or more lasers of specific wavelengths, and these are used to excite the electrons in the fluorochromes, energizing them, which then results in them emitting light, which can be picked up by the next part of the cytometer, which is the optics. So flow cytometry optics, which you can see a schematic of here, are a series of dichroic mirrors, bandpass filters, and PMTs, or photomultiplier tubes. And before we go any further, we should probably discuss what all of those things are. So dichroic mirrors, which are the first part of the optics, are essentially beam splitters. And what this means is that they will reflect certain wavelengths of light while allowing other wavelengths to pass right through them. So using a dichroic mirror, the cytometer is able to sort out light which has been emitted by different fluorochromes. So a dichroic mirror will typically allow all light below or above a certain wavelength to pass through. For example, this 550 LP or 550 long pass mirror here, this will filter all light over 550 and allow it to pass through, but it reflects everything below 550 nanometers. So this reflected light then next goes through a bandpass filter here. These filters allow a range of light around a certain wavelength to pass. For example, this 530/10 would allow light of a wavelength 530 nanometers plus or minus 10 nanometers to pass through. The light then reaches the PMT, or the photomultiplier tube. The PMT is struck by the photons of light, and it can convert these photons to electrical signals, and the software then can interpret these electrical signals and decipher them into dots on your dot plots. So that's really the very basics of the principles of flow cytometry. Obviously, there's much more information available in different resources, but I think we've covered the fact that there are many different types of flow cytometers, but all of them work on similar principles. Flow cytometry can be used to measure physical characteristics of cells, such as size, granularity, and any fluorescence. And it uses lasers, fluidics, and optics to do this. So next, we'll move on to talk about sample preparation. And with sample preparation, there's one big take-home message, and that is that single cells are key. If we remember how the cytometer works, which we can see again here, it's all based on single cells. To get the best quality data, we need single cells flowing through our flow cell and being interrogated by the laser one at a time. Doublets or triplets or clumps of cells are no good to us. So cells for flow cytometry can come from a wide range of different sources, such as organs, and here I've just used the lungs as an example, tissues like skin biopsies, from cells in culture, or from bodily fluids like blood. But no matter where your cells come from, you need to get them into a single cell suspension. But how you do this can be very different for different cell sources. So to prepare solid tissues like organs or tissues or even 3D spheroids from cell culture, there are different approaches which can be taken. So you might want to use a mechanical or enzymatic approach to break down the tissue into a single cell suspension. For some tissues like the spleen or lymph nodes, mashing the cells through a cell strainer using the plunger of a syringe is very effective. But for other tougher tissues like skin, enzymatic digestion might be necessary. There are loads of different protocols out there for digestion using enzymes. You can use proteases, trypsins, acutase, and there's loads of others. Cultured cells then are most commonly removed from their flasks enzymatically using trypsin or acutase. But sometimes people will scrape cells off, use EDTA or other mechanical means. All methods can cause some cell death. You need to be careful, and the most important thing is you need to be consistent with your method. For blood or other fluids, there are some considerations before you stain and run your cells. So do you want to do a gradient centrifugation on your blood to separate the white blood cells from the red blood cells? Do you want to lyse your red blood cells to exclude them from your analysis? And there are loads of different products available for this. Through MSC, we supply red blood cell lysis buffer from BioLegend, and also a lymphocyte separation medium to do your gradient centrifugation from Tonbo. And you can talk to any of us, to Lisa, Emma, or myself, and we'll be happy to chat to you about those products. The key thing though is whatever protocol you decide to do is to optimize it and to make it consistent for your experiments. So when you're optimizing your protocol, there's a few things you want to look at. You want to minimize the amount of death in your cells. You want to ensure you're getting a consistent single cell suspension across all of your experiments. And you want to ensure that your protocol does not alter the markers, the expression of the markers that you are interested in. Another tip is to always strain your cells through a strainer before putting them on the cytometer. This is to avoid blocking the flow cell with clumps or with extracellular matrix. And you want to keep your cells cool throughout the staining process, the preparation process and the staining process, to ensure that you're not changing the marker expression on your cells. So next we'll move on to chat about how you'd stain your cells. So once you have a single cell suspension, the next step is to use fluorochrome conjugated flow cytometry antibodies to bind to the markers of interest on the outside of your cell. This is to allow you to run your cells through the cytometer and find out how many of your cells have your marker or your markers of interest on them. So staining cells involves incubating them with an antibody against a cell surface protein. Generally, 95% of the time, this is done using the direct method. So this is where the antibody you are using is directly conjugated to a fluorochrome. And therefore, you just allow time for the antibody to bind to the cell surface molecule. Then after a wash, you're ready to go on the cytometer. Sometimes though, a primary and secondary antibody setup will need to be used in the indirect method. So here you have a non-conjugated primary antibody, which you can see here in green. And this is allowed to bind to the cell surface marker. Then you come along with a secondary antibody, which is another antibody against the primary antibody. This secondary antibody then is conjugated to the fluorochrome. So when choosing fluorochromes, there are a number of different parameters to take into account. So you need to remember that each fluorochrome has distinct properties and it's characterized by specific excitation and emission wavelengths. So first, the fluorochrome must be excited by the lasers you have available on your instrument. And secondly, the emission wavelengths must be detected by the detectors or photomultiplier tubes on your instrument. That's very important. You must also think about the brightness of the fluorochrome. So using the brightest fluorochrome, which is typically PE, should be used on a protein that has the lowest expression level, and vice versa, the dimmest fluorochrome should be used on the most highly expressed protein. You should think about spectral overlap and try to avoid it if possible. So fluorochromes that have a minimal amount of emission spectral overlap generally are further apart on the emission maxima, and they will have less spectral overlap, which is going to make your compensation easier later on. There are many resources available online to help you with panel design, and I'd thoroughly recommend that you check those out. So the buffer then is also very important. FACS buffer generally contains these four components. So PBS, fetal calf serum, EDTA, and sodium azide. The concentrations can sometimes vary a little, but it's generally these four components. And the EDTA, as we all probably know, is there to limit cell-to-cell interactions and reduce cell clumping, helping you have a single cell suspension. Sodium azide helps to prevent the internalization of surface antigens. And this, in combination with doing your staining at low temperatures, like 4 degrees, will help to maintain the surface profile of the cells. This is to ensure consistency. And then if you want to, we do sell a flow cytometry staining buffer from Tonbo BioSciences. It's already produced in a quality controlled environment, so you can ensure consistency across all of your experiments. There's some tips then for working with your antibodies. So all of your antibodies should be titrated to ensure that you're using the minimum volume to stain all of your cells. This avoids the effects of excess antibody in your solution. You should also be consistent with the cell number you're using across your experiment to ensure consistency. Generally, between 1 and 5 million cells per mil is a good guide to start off with. So I just wanted to give an example to you today of a 3-color experiment. What controls you need to have to set up the cytometer, do your compensation, and set up your gates. Of course, as we said, you should use the same number of cells in every tube. You should stain for the same amount of time with the same amount of antibody in the same volume of FACS buffer. Now, for today, we're going to assume that we have FITC, PE, and APC, and these antibodies are conjugated to CD3, CD4, and CD8 respectively. Now, you could be using any cells here. We could be using blood cells. We could be using splenocytes. We could be using lymph cells. But obviously, it's immune cells that we're looking at with these markers. So here are the controls that you should have for this 3-color experiment. So you should have an unstained control, which is cells with no antibody at all. You need then single stained controls, excuse me. So you should have a single stained control for each cell type. So you need a CD3 only, a CD4 only, and a CD8 only. Then you need fluorescence minus 1 controls, or FMOs. So these are cells which are stained with 2 out of your 3 antibodies. If you had a 4-color experiment, it would be cells stained with 3 out of your 4. So you're missing 1 every time. So we would say that you've got your FITC FMO would be cells which are stained with your PE and APC antibody, but not with your FITC, and so on for your other fluorochromes. Another thing to consider is whether you're going to use isotype controls. This is to be recommended, and isotype controls are there to limit the unwanted background cell staining, which can be a problem in flow cytometry. So this is especially a problem when you're detecting novel or very rare populations, or when you're working with a panel that has multiple fluorochromes on it. So an isotype control is an antibody which is raised against an antigen which isn't present on the cell type that you're looking at. This antibody control has been specifically developed to determine background staining. So it will help you figure out if you've got non-specific binding of your antibody to FC receptors, which are found on lots of immune cells. It will help you ensure the staining you're seeing is due to specific binding rather than an artifact. And it will also reveal if you've got other non-specific binding of the antibody or the fluorophores even to other cellular components or extracellular matrix or anything that might be there. So next, we'll move on to talk about fixing cells and when you might fix or why you might fix cells. So we'll start off with why would you fix your cells? So there's several good reasons to fix cells for flow cytometry. The first is if you want to stain intracellular targets such as cytokines or phosphorylation of transcription factors, for example. And if you need to permeabilize the cells to do this, you need to have the cells fixed first. The second reason is convenience. So depending on your experiment, you may have a really long day preparing your cells and getting them ready to stain. And if you don't fancy sitting on the cytometer until midnight, sometimes it's better to fix the cells and leave them in the fridge overnight so that you can run them the next morning. The next reason is disinfection. If you're working with dangerous infectious samples like, say, HIV infected cells, it's routine to fix them prior to your analysis so that you don't contaminate the cytometer with a dangerous pathogen and place yourself or others at risk. Another reason is if your experiment involves time points. So if you're looking at a progressive biological process over time, for example, the phosphorylation of a protein in the hours after a stimulus, you might want to fix all of your samples at those time points and then analyze them all together later as one experiment. The next question to ask yourself is which fixative should you use? By far the most common fixative is paraformaldehyde. This can be made up yourself or you can purchase some from us, such as the BioLegend fixation buffer. The time needed to fix your cells with PFA can vary. You see different protocols online from 10 minutes up to 45 minutes. And again, like anything else, you should optimize this yourself. There are other fixation buffers, which you will see. They have different active ingredients like formaldehyde, alcohols, etc. Another big question is when should you fix? And by this I mean, should you fix your cells before you stain your extracellular markers or afterwards? The answer to this is there is no answer, and it depends on your experiment. So for example, if you're looking at the phosphorylation of an internal protein, you might want to fix before you stain your extracellular markers. And this is so that the phosphorylation state of that protein you're interested in doesn't change in the time that you're staining your extracellular markers. However, though, if you're looking at a cell surface marker whose epitope is changed by the fixation, you might want to stain first so that you aren't missing any of the marker that you're interested in. There's a really good blog on the BioLegend website, which I'd advise everybody to check out, and this discusses when and how to fix in a lot more detail. So now I'm just going to, this is sort of FOXP3 intracellular staining protocol, which I'm just going to show you, just to show you how simple intracellular staining can be. So using the FOXP3 fixed perm buffer set from BioLegend, all you need to do, you get your cells, you perform your surface staining as usual, add your fixation permeabilization solution to the cells and incubate for 20 minutes. You just remove this buffer then, wash your cells with FAX buffer, wash them with the permeabilization buffer. Then you resuspended permeabilization buffer, which has your FOXP3 antibody inside, and incubate for 30 minutes at 4 degrees. And again, just spin down your cells, wash, and you're ready to go on your cytometer. So it's a very straightforward, simple protocol, which can be optimized quite easily for any lab. So now we'll move on to setting up your cytometer once you have your cells stained and they're ready to go. So if we come back to this image again here, we know that a laser will excite your fluorescent molecule on the cell as it passes through the flow cell. This light is then filtered and captured by the PMTs for each channel. But what happens next? So basically, how does the photon of light hitting the PMT end up as a data point on your plot? Well, once the light signals or the photons strike the PMT, they're converted into a proportional number of electrons. These electrons then are multiplied and create an electrical current. The electrical current travels to an amplifier, which converts the current to a voltage pulse. The size of the pulse depends on the number of photons which were detected, but also depends on the voltage of the PMT. So you can further amplify signals by increasing the PMT voltage. And this is a method of allowing you to detect weaker signals, so very rare markers or weakly expressed markers. So when you're setting up your voltage, you should first run your unstained or your isotype control and cycle through the channels and set the voltages of your PMTs so that your unstained cells are at the left-hand side of the x-axis. This leaves plenty of room to the right for your positive cells to come up later on. Then the next thing you want to do is run your stained control, your single stain control. For example, in this case, we're talking about FITSI. And ideally, you will see a clear separation between your unstained and your stained control. You then go along and repeat this for all of your other channels. But you need to remember, once you set the voltages, they must remain the same for every tube in the whole experiment. Otherwise, your data will not be valid. So next, we'll talk about compensation. So compensation is almost always necessary in multicolor flow cytometry experiments. And it's a mathematical process by which we can correct multi-parameter flow cytometry data for spectral overlap. So we all know what spectral overlap is at this stage, but just to recap quickly, the emission spectra of many of the fluorochromes overlap with each other. So for example, you can see here that the FITSI emission spectra and the PE emission spectra overlap quite a bit. So this means that some of the fluorescence seen in the FITSI channel is actually emitted by PE and vice versa. So we need to have a mechanism in our flow cytometry software to deal with this, filter it out, and then we can only look at the fluorescence generated by that fluorochrome that we're actually interested in. So to do compensation, you need your FMOs, or your fluorescence -1 tubes. So we'll say, for example, that we're using our FITSI FMO. So that's a tube of cells, again, stained with only our PE and APC antibody in our example experiment. So we set up a dot plot with FITSI on the y-axis and PE on the x-axis. And when you run the cells uncompensated, you see that the PE negative population looks nice down there in the bottom left-hand corner, but you can see the PE positive population looks to be also FITSI positive. And the more positive the PE is, the more positive the FITSI seems. But we know this is impossible because we've got no FITSI antibody in here, so it must be spectral spillover. So that is that the PE fluorescence is showing up in the FITSI channel. So this needs to be corrected. So in your software, there'll be a place where you can adjust the compensation between channels. And in this case, you need to adjust the FITSI PE compensation until the PE positive population has the same FITC fluorescence as the PE negative population, which we can see here in this compensated data on the right. You then need to complete this for all of your other FMOs. And similar to voltages, once your compensation settings are set, you need to keep them constant for all tubes in your experiment, otherwise the data is invalid. Now, of course, most cytometers and analysis packages have automatic compensation modules now. This automates the process, makes it a lot more standardized and a lot more straightforward. But I just wanted to go through the theory behind it here, because some people obviously still do manual compensation, as far as I know. So now next we'll talk about data analysis. So once you've acquired your data on the cytometer, it's saved as a Flow Cytometry standard, or FCS file. So this is a standard file which was developed by the Society for Analytical Cytology. And the file contains a description of the sample, the instrument on which it was collected, the raw data itself, any analysis which has been done up to that point. Now, typically a single cell analyzed for 4 parameters will generate about 8 bytes of data. So if you measure 10,000 cells, each FCS file will be about 80 kilobytes. So you can see that over time, storage space is also an important concern for flow cytometrists. So once your data is saved, there are different analysis packages which you can use to look at it, delve into detail of the different populations, etc. So there are loads of different ways to visualize your data, but today I'll just focus on probably the 2 most common. And this is the histogram, first of all, here on the left. So in the histogram, the fluorochrome is on the x-axis. And here it says FL1, but you can change this to FITC or PE or APC or whatever your fluorochrome is. So the x-axis measures the intensity of the fluorescence, and it's often displayed in an exponential mode, as there can be a very large difference between negative cells and very strongly positive cells. So an exponential axis is often easier to visualize. On the y-axis of the histogram, we generally have a cell count. So the histogram then represents the number of cells at different fluorescence intensity levels, from very low fluorescence here on the left, which are essentially negative, to strongly fluorescent over on the right-hand side of the histogram. The second commonly seen plot, which we have here on the right, is the 2D plot, or the dot plot. So here you'll have one channel on the x-axis and you'll have another channel on the y. The channels can be fluorescent fluorochromes, or they can be forward scatter or side scatter, or a combination of both. So this allows you to distinguish different populations based on the differential expression of markers or on physical parameters such as forward scatter and side scatter, looking at size and granularity. So this allows you to distinguish between, say, lymphocytes and neutrophils in our previous example, or it allows you to distinguish between 2 different markers. So, for example, CD4 and CD8 T cells would be quite a common one, which would look quite like this. So the next part of analysis is gating. So a subset of data can be defined through a gate. So a gate is a numerical or a graphical boundary which can be used to define the characteristics of particles which you want to include for further analysis. For example, a blood sample containing a mixed population of cells, you might be interested in restricting your analysis to only the lymphocytes. So based on forward scatter and side scatter, you can set a gate to allow analysis only of the cells of the that are in the lymphocyte area. The resulting display then would reflect the fluorescence properties of only the lymphocytes. So you can gate on both the dot plot and the histogram. You can see here that we've gated on the negative population and the positive population of the CD3 FITC marker here. And gating then is where your controls, especially your FMOs, come back into their own. So when you use FMOs, you establish scientific evidence as to why the gates you drew are drawn correctly. So this is true for any multicolor flow cytometry experiment. So FMO controls will give you, as well as anybody who's looking at your work, confidence in the degree of accuracy of your measurements. So you can see this represented quite nicely here. So on the left, we have an unstained control. You can see that if you were to use just above the negative population as where to draw your gate for this CD45 RA positive, you'd get a very different result than if you use the FMO, which is in the center here. Because all you want to see is a genuinely PE positive population and not any stray or spillover light from other fluorochromes or from autofluorescence, you should use the FMO to draw your gate. What you can see here is made a massive difference between the red and the blue line, but the blue line is the correct location to draw that gate, and you're getting the correct information here. There are many ways to display flow cytometry data in either presentation or publication form. The first and probably the most common is to display percentage positive cells. So this can be a percentage of the total cells analyzed, but more than likely it's a percentage of some parent population. So for example, you might say that 30% of the lymphocytes you analyzed in a particular sample were CD4 positive, or that 5% of the F480 positive macrophages were also CD169 positive. So this gives you really useful information, particularly if you're examining the effects of a treatment on cells. A flow cytometer can also be used to determine the absolute number of cells in a sample. For example, CD19 positive B cells per milliliter of blood. There are different ways to get absolute numbers like this, but among the most common and the most reliable is to add a defined number of counting beads to each sample and to use these beads to standardize samples between each other. Some cytometers now can also measure the volume of fluid going through, and this can be used to obtain absolute numbers as well. Another very useful measurement is looking at the intensity of fluorescence. So this is sort of similar to a Western blot, where you can look at relative intensity levels between different samples. It's particularly useful for activation markers, such as CD25 on T cells, or intracellular markers like transcription factors. So data analysis packages will give you an average fluorescence intensity for the cells in a particular gate. And you can use this to compare between different samples, which might have received different treatments, for example. So just to wrap up our session today, we have seen the very basics of how a flow cytometer works. We've looked at some basic tips on sample preparation, staining your cells, fixing cells. And we then moved on to setting up the cytometer to run your experiment and some introduction on the principles of data analysis. There are loads of really good resources online, including PDFs from universities, YouTube, suppliers' websites and blogs, where you can get really good explanations of some of the principles behind the things like compensation and gating, as well as some really handy practical tips to make life easier day to day in the lab. From my sort of 7 years of flow cytometry experience, if I was to impart one piece of my own advice from my experience, it'd be to standardize your protocols at the beginning of your project. So for your sample prep to staining to analysis, standardize everything, optimize it all. So, and then you get the best quality data out at the end, and you actually save time by not having to repeat experiments. So just here are just some of the references that I have used in preparing today's talk, which will be available to you. You can download the slides. And finally, I would just like to introduce Tonbo BioSciences. So Tonbo are a new supplier for us here at MSC. They were founded by a team of immunologists in 2011, and they're based in San Diego. Their focus is on delivering the same high quality reagents as other suppliers at an affordable price. Their prices are really reasonable, and they also have a very wide range of pack sizes available. This is a screenshot directly from the Tonbo website. And as you can see, they supply a broad range of reagents, from antibodies and other flow cytometry reagents to recombinant proteins, to ELISA kits and molecular biology reagents, including cloning kits. And as always, if there's something on the website you're interested in, please get in touch with either Lisa, Emma, or myself, and we'd be very happy to help you out. As you're all aware, there's a discount for attending and listening to me drone on today, which can be used for any flow cytometry reagent from our portfolio. We will send you out an email with that discount code, and you can contact us for a quote. And finally, just if you've got any questions, and I'd like to say thank you all for listening. And you can see Lisa, myself, and Emma's email addresses here if you need to contact us for anything. So thank you very much for listening today, and I hope you enjoyed it.